Hi, I'm Kate Thurlow,
And I'm Owen Evans.
and we're here to give you a short yet precise account on the evolution of life on Earth from pre-RNA to the first cell. Primitive Earth hosted conditions unfavorable to sustain life. Unstable conditions caused by high temperatures, continual waves of ultraviolet and cosmic radiation. This early Earth had no water, a fundamental component to life as we know it.
The hydrosphere formed through mantle differentiation 4.6 billion years ago. This led to the formation of what is known as the primordial soup. The primordial soup environment was created in the Miller-Urey experiment and successfully produced 17 of the 20 known amino acids.
The production of amino acids in this environment allowed peptides to form. Peptides are chains of amino acids that are the basic building blocks of proteins and single-strand ribonucleic acids. This characterizes the pre-RNA world, where simple proteins and ribozymes are formed. These simple proteins are the foundation for ribosomes, which are organelles that sequence amino acids for gene expression.
Ribozymes are RNA enzymes that cause catalytic conversion of peptides. This is fundamental to the movement from the pre-RNA world to the RNA world, which is characterized by the ribozyme.
The presence of ribozymes led to the building of more complex proteins and the transcription of the first code in RNAs. This code dictated the genetic expression of the RNAs and through genetic fidelity how the M, T, and R RNAs evolved. This code led the way to the formation of ribosomes.
Ribosomes, which are the site of translation, are made up of complex proteins and RNAs. These proteins are called ribosomals, which are special because they can read and translate RNA, which is a step that is critical to the formation of DNA's double helix. The movement from the RNA world was characterized by the formation of DNA.
DNA formed through the alteration of a self-replicating RNA genome. This self-replicating RNA was created through the interaction of the ribozyme environment with the complex RNAs. This in turn formed a template for RNA to self-replicate. The rigid structure of the double helix formed as a result of the continual collapse of the single-strand RNA. This cell formed in stages beginning with the creation of the cell membrane. This occurred when peptides containing 4 to 10 hydrophobic residues self-assembled to form nanovesicles. The formation of these nanovesicles was a semi-random synthesis which increased internal concentrations of hydrophilic peptides and nucleic acids. Nearby lipids that formed through early chemical processes were attracted to the hydrophobic regions of the membrane's peptides, which led to the formation of a hybrid cell membrane.
These primitive vesicles host RNA, DNA, and ribosomes. The ribosomes are present due to the collapsing and reformation nature of the internal peptides. This, coupled with how the vesicles form around ribosomes, means that these organelles were present in the first cell. The DNA within the protocell starts to balance and coordinate with the RNA and the ribosomes. The DNA uses the mRNA to instruct the ribosomes to form specific amino acids, which eventually leads to a stable, simple cell.
Once this simple cell is finalized and stable, it can be replicated, leading to all complex life on Earth.